I'm really happy to be talking today to Brittany Walters to discuss her recent PLOS ONE Lab Protocol paper entitled Longitudinal Imaging of the Taste Bud in Vivo with two-photon laser scanning microscopy and the associated detailed protocol in protocols.io. Brittany is a fourth-year PhD candidate in anatomical sciences and neurobiology at the University of Louisville School of Medicine. Her research focuses on the cellular dynamics of taste buds, where she uses in vivo two-photon microscopy and genetic labeling to track individual taste buds over time. In doing so, Brittany is able to capture the lifespan of different taste bud cell types with the goal of understanding how the morphology of these cells changes throughout their lifespan. She holds a master's degree in anatomical sciences from Lincoln Memorial University and a bachelor's degree in biology from Covenant College. Her work is currently supported by a two-year NIH Diversity Supplement Award through the National Institute of Deafness and Other Communication Disorders. Brittany, thank you so much for carving some time to talk to me today.
Yeah, it's great to be here. Thank you so much for having me.
So to kick off, can you briefly explain what this protocol is about, how to use it, and why it is relevant?
Yeah, absolutely. So this protocol is all about how to repeatedly track taste buds on a mouse's tongue over time. I think this method could probably be adapted to more than mice. It just so happens that our lab uses mice. And so this method was a great addition to our field because while other laboratories had been able to demonstrate live imaging in taste buds in mice, we were the first to demonstrate the feasibility of repeatedly identifying and imaging the same taste bud over time. And I have an image here just because I think it's a bit difficult to actually understand what's going on. And so here you can see that the mouse is anesthetized and we pull its tongue out from its mouth using forceps. And then we lay like a cover glass holder down over the tongue so that you can see like the anterior tip of its tongue. And then you can image just like that. And I like to include this image just because I feel like it's a bit of a difficult process to explain. But yeah, that's what it's all about. Just repeatedly imaging these taste buds so that we can analyze individual cells, how they change over time, and get a better grip on what their lifespan is and how their morphology changes as they age in the taste bud.
And that was a pretty cool picture. Now I understand the setting.
Okay, good.
But, you know, and to better appreciate the significance of your optimization and the development of this protocol, can you briefly elaborate what was the state of the art? So were people not imaging longitudinally the taste buds in rodents before?
That's correct. Yes. So this is the first method which allows you to longitudinally image taste buds across time. So we've imaged taste buds up to like 60 days or 100 days in a row without any damage to tissue using two-photon microscopy, as you said previously. And yeah, it's great because we can track dynamic processes like our paper that came out in PLOS Biology a few years ago with you guys. We demonstrated the dynamic turnover of terminal arbors as those nerve fibers connect to cells. And we were really surprised that nerve fibers, we call them arbors, turn over in less than 6 hours. It was really pretty incredible. It just speaks to how dynamic structures in the taste bud are always changing, which makes sense because our tongue it faces a lot throughout the day, right? Like we're constantly eating, drinking, like something. And so yeah, this has been a good approach to allow us to image and visualize those dynamic changes in the taste bud.
And in addition to the ability of imaging during a long period of time, are there other advantages, unique advantages of this protocol over alternative approaches?
I think absolutely. Like, so not only can you image individual cells, track their lifespan, track how their morphology changes, you're also able to fluorescently label with stains. Like we use the Hoyt stain to label cell nuclei, and we use a mitochondrial stain to label mitochondria, which we demonstrate in the paper. And so we hope that folks will adapt this protocol and use different fluorescent labeling techniques to image organelles and see how those are also changing as cells age in the taste bud. So we hope that this protocol is like a baseline and that others can adapt it for their needs.
And now we're going a little bit deeper into the developing of the protocol itself. Can you tell us a little bit about the highs and lows of the process? For example, is there anything that you tried as you were developing the protocol, but didn't work, and therefore it didn't make it to the final version of the protocol?
Yeah, absolutely. So developing this protocol was like one of my PI, Robin Krim's goals for her career. And so she collaborated with Aaron McGee and Zachary Whittem, which was a graduate student of hers at the time, to develop this method. And I am the receiving graduate student who learned this method from Zach. And so it was really great to kind of like walk into this new method that our labs had collaborated and developed together. And yeah, there were definitely, I heard a lot of troubleshooting errors that they had. They initially discovered or realized that this was possible because a lab at Harvard demonstrated use of a suction grabber to actually pull the mouse's tongue out over time and image, not longitudinally, but just image. And we adapted that method. We tried using like clay to like stick the mouse's tongue to. We tried using tape and other things. And I even heard that they at one point purchased a tattoo gun to try to like tattoo an X on the mouse's tongue, be like, okay, X marks the spot. This is where we're going to go to try to find the same group of taste buds, because that's how we do it. We take an initial papilla map of the mouse's tongue, like on day 1 of imaging, because taste buds don't move around on the tongue over time. They're standardized in their positioning. You can repeatedly go back to that papilla map and use it day after day after day to find your group of taste buds that you're imaging. And so that's why they thought that having a tattoo gun and labeling an X on the tongue would help them identify that location that they needed to go back to find that same group of taste buds. So there were definitely some troubleshooting that was done that I've heard about in the lab. But ultimately, this method was successful. Zach, I think the first time he created a tongue holder, he used a USB backing. You know how they have like the metal covering around the USB itself? He actually used that metal to create this circular tongue holder, and then cut out the middle portion and inserted the cover glass holder. So he was very creative in developing this method, and yeah, it's worked really well for our lab.
And when people use the protocol, what are the most critical steps? What parts are particularly tricky and others might make a mistake on?
Yeah, so I feel like pulling the mouse's tongue out of its mouth, it's a very small mouth, repeatedly over time is very tricky, because you're trying to get it in the same position on top of that metal bar each time so that your group of taste buds is the same. Otherwise, you might see like half of your imaging field, but it's going to be really, really hard to line it up to your original image on day 1 of your papilla map. Be like, oh, okay, this is the same taste bud that I was imaging before. So it's really important to position the mouse's tongue the same time day after day for imaging. I think that because the mice aren't restrained, anesthetizing them and keeping them anesthetized at a consistent level is a bit difficult. They're just laid on the imaging platform and their tongues fold out, so there's no like restraining of their limbs. So yeah, consistent anesthesia so that the mouse is anesthetized and calm Otherwise, its tongue will jerk, which will cause all kinds of motion artifacts for imaging.
And at the beginning of our conversation, you briefly mentioned that the method, the protocol was developed for mice, but that it should be easily expandable to other rodents
Yeah.
or animals. Have you ever tried to apply this method to rats, let's say? And if not, what do you think will be the required optimizations to have this method work in other model organisms?
Yeah, so we have not tried in rats, mostly because we're not approved to work with rats in our laboratory. But I do think that it is definitely adaptable. Everything would have to be enlarged, obviously. But sorry, can you repeat the last part of the question?
Yes, what optimization do you think people have to do to make these to work in other model organisms, like rats, for example?
Like what other things would be necessary
To optimize?
To optimize? Yeah, that's a really great question. I would say like the imaging platform that I described how to make using different parts. I would say that would definitely have to be expanded. I would say that it would probably be best to have the rats restrained just because they are larger than mice. I would also say that you might need to limit your imaging time so we can image like about an hour without any issues. My predecessor, Zach, could keep mice down for much more hours, but we try to really limit how much anesthesia they do receive. So I try to keep my imaging time points to less than an hour. But I would say that you would really need to watch how long you can actually image in the rats for. So yeah, hopefully that answered a bit of the questions.
Absolutely. And you already also touched a little bit on this, but what kind of experiments or application has this protocol enabled that weren't possible before?
Yeah, absolutely. So because of this technique, we're now able to label with GFP or with TD tomato individual cell types in the taste bud. And so we're able to track cells saved from initial entry into the taste bud all the way to apoptosis, cell death. And so we can visualize how their morphology changes. They kind of initially come into the taste bud and they're little round globs, but then over time, the taste bud cells elongate, become more fusiform in shape. And so because we can track individual cells, we can better define how morphology changes and particularly look at if subtypes of cells do exist within, say, like the type 2 cell population or the type 3 cell population based upon differences in morphology. And so it's allowed that, as well as what was discussed in the PLOS biology paper was nerve fiber remodeling. And obviously this method has allowed us to look at those super quick dynamic changes of how those arbors change over time, oppose cells, and then kind of like back away and remove themselves from cells, perhaps as the cells are exiting the taste bud and undergoing apoptosis. Yeah, lots of new exciting things, which I will kind of branch out on my own here just for a second. But because there's all of this, because of this new method, there's a lot of new data. And the field doesn't traditionally have a lot of quantitative methods to analyze large scale volume data sets like this. And so because of that, I went to my boss, Robin, one day and I was like, hey, it's really tedious to track these taste bud cells manually by hand, especially in the type 2 cell population, because those cells are really densely packed in the taste bud. And so trying to track them from day to day is super tedious manually by hand. And I was like, do you know if there are any automated methods to track these cells just to like speed up how much I can accomplish in a day? She's like, I really don't know. But why don't you apply to this course at the Marine Biological Laboratory? And I was like, okay, great. And I did, and I was accepted into the course, which is great. And there I was exposed to all different types of tracking methodologies like motile and ULTRAC to actually, which are automated softwares, which can segment and then track those taste bud cells over time. And so I'm currently working with a computing research engineer in our department right now to facilitate automated segmentation and tracking of those taste bud cells. This is a project I'm really, really excited about, because with this automated segmentation, we can render these beautiful segmentations and really, really see the structure of these cells as they do progress from post-mitotic precursor cells in the bud. Those are like newly entered cells all the way to cell death. And so this project really has expanded our ability to visualize cell morphology. And yeah, I'm really excited about it. So it's been really great, I'd say, for my career in exposing me to different imaging techniques, and then also to different ways to quantitatively analyze your data. So, yeah, I know I kind of went off on my own there, but
no, that's very cool, Brittany. I would like us now to change gears a little bit and talk about peer reviewing the protocol. So this method, as I understood, and you briefly mentioned it as well, was used in a couple of publications before, including one by your own laboratory in PLOS biology. What was then the motivation to send that protocol to PLOS ONE to be peer-reviewed as a standalone methodological paper?
Yeah, so we really saw the value in just getting the method out there and presenting it to the field. We would really love and have seen other laboratories already incorporate and use this method. And so, yeah, we wanted to have it out there. Yeah, just to allow folks to adopt it and adapt it for their own unique studies. So that's why we wanted to go ahead and get that out to PLOS ONE and publish with you guys, which is great. We were able to. And the review process was really, really smooth, and I had a good experience. There weren't a lot of comments on the protocol itself, but there was a lot of good feedback about how to revise the text, which in turn made the protocol more clear, which was very important. And so revision from the reviewers, it was a good experience. And we really feel like some of the suggestions really improved what we were able to present.
And just to finalize, this is my last question.
Yeah.
What has been the significance of publishing this lab protocol paper for your career as an early career researcher?
Yeah, absolutely. So I think I touched on that a bit earlier when I went off on my own, but it's been really great because not only has it exposed me to this new technique, which I can bring to the field with my lab, but also it's really exposed me to different ways to analyze data, which has been really important. Because like I said, these data sets that are generated, there's just a ton of data, and there's a lot that you can pull from the images. And so to be able to improve my computational understanding and understand how to like extract data from these images has been really, really useful.
Thank you. As I mentioned, I don't have any more questions.
Okay.
But before we wrap up, I want to know if there's anything else that you would like to mention that we haven't discussed so far.
No, I think this is good. Yeah, this has been great.